All right, welcome to the next video of General Science. And in the last video, we talked about DNA and its structure and kind of what it does. And now what we're going to talk about is how that DNA replicates, like we talked about in our cell division video. So to start off, let's talk a little bit about how we figured all this out. In 1962, the Nobel Prize in Physiology and Medicine was given to James Watson and Francis Crick and Maurice Wilkins because they figured out what the molecular structure of DNA was. Now, there's some controversy about that because they ended up taking a bunch of data from Rosalind Franklin, who actually was the crystallographer who got the data ready for them, and then they made the conclusions, and so they got the Nobel Prize and she didn't. It's one of many examples of women being kind of shoved to the background while men took the glory for scientific achievement. So like we said in the last video, the structure of DNA is composed of the 4 nucleotides, adenine, cytosine, thymine, and guanine. And the amounts of A and T's and G's and C's create this double helix. And so whenever you see one nucleotide, the opposite is going to be across from it. And it forms this double helix pattern. And so the relationship between the structure and the function is manifest in double helix. And since the 2 strands of DNA are complementary to each other, they can act as a strand and a template to build a new strand. So how DNA replication is going to work is that parent molecule is going to unwind, and now there's 2 new strands that are opposites of each other, right? So in our first one here, we've got ACTAG and TGATC, and when they split across, if you put the new nucleotides in, you're going to end up with 2 identical copies. So that's how DNA replicates itself. By splitting in half, it can turn itself into 2 identical copies of the original. Now, DNA replication is what's known as semi-conservative, right? So that means each new strand is only going to be half new, right? So there's going to be one strand with the original stuff and the other strand without, and so it's not always completely new. The copying of DNA is pretty remarkable because it's pretty quick and it's accurate. It involves unwinding this double helix and synthesizing 2 new strands. And there's more than a dozen enzymes and other proteins that participate in this process. But the replication of DNA begins at special sites called the origins of replication, and that's where those 2 strands will be separated. So basically what will happen is in this origin of replication area, it's going to begin at these specific sites, and it's going to separate them, and it turns into what's called a bubble. So if you take a look here in this picture over here, you can see the arrows. That's where that bubble is forming. Okay. The bubbles expand laterally as the DNA replication proceeds in both directions, and then eventually the replication bubbles fuse and the synthesis of the daughter strands is complete. So it's never actually made all at once. There's usually like several different groups of areas that are being replicated at once, and they eventually end up being a complete new strand. So to start DNA, it's catalyzed by what's called a DNA polymerase, and it needs a primer. So what happens is a primase, which is a different type of protein, adds the primer onto the DNA strand, and then the polymerase adds nucleotides to that primer and continues on, as you can see in this picture here, right? The primase has added the primer, and then this DNA polymerase is adding new nucleotides and making that new strand of DNA. The nucleotides are added by complementary base pairings, just like normal, and then there are substrates and different proteins that are helping release the energy for all this to happen, making sure all the connections can be made. So there's lots of different proteins doing different things in this. DNA synthesis on the leading strand is continuous. So on the top strand there, you can see that's called the leading strand. And what happens is because it's going the correct direction for the polymerase, it's going to make this whole thing here all at once. There's not going to be any stops or anything. However, the lagging strand, because it's not going the correct direction, because remember it's the opposite of the other one, so to make it work, basically what happens is it's discontinuous. So the DNA is added in short fragments, which are called Osaki fragments, and that's named after the guy who discovered this. And so what happens is the DNA polymerase will make small sections, and then there's another protein that will connect those segments together. So one side is a lot more smooth. The other side has to be discontinuous because of the direction of the DNA. So here's another example of how that would work. So on this side, right, it's going the right direction, so it's all just created all at once. And then on what's called the lagging strand, they have to make these Okazaki fragments, so they're going to make chunks, and then they have to be connected together. So that part takes a little bit longer than the other one. There's some arrows there. Right, because the overall direction of replication is this way, but because this one has to be bonded this way, it has to be done a little different. Then at the end, there's another polymerase that degrades the primer and replaces it with some more DNA, and you now have a complete daughter strand. Remember, that primer is not part of the DNA strand, so it has to get cut out and then replaced with the actual DNA at the end so that it is actual DNA. There are so many proteins that assist in this process. There are helicases, which unwind the helix. There's single-stranded DNA that's binding proteins and making the template available. There are different primases that make the primers. There's polymerases that do all of the creation of the new DNA. There's also a polymerase that gets rid of the primer, and then there's a ligase that combines all the different fragments on the other one. So there's lots of proteins that are involved in this process. It's a huge undertaking. Here's some pictures of what all those enzymes are going to look like in our little showing of them. And so one more time, we'll go through the structure again. So we've got our helicase here, and it's unwinding the DNA. So it's basically unzipping it. And then we've got some binding proteins that are going to hold these in place so that they don't try to reconnect. And then they're putting on their primers. So that's these red guys. So they're putting their primers on those green sections. Now we're replicating going this way, so this strand is going to be the continuous strand because it's going the right direction, and then this one's going to be the discontinuous one because it's going not the same direction as the replication direction. So they're going to keep adding on, and as you can see, this one's adding on nice and quickly, but this one, remember, is going to be making those fragments. So it made a fragment, and now it's going to move back and make a new fragment. The discontinuous synthesis is going to produce all those Okazaki fragments while the leading strand gets to make its full thing. And then we're going to remove some of those primers. And then there's a ligase that makes the backbone correct, and the DNA polymerase fills in some gaps. And we end up with a full set of DNA. Now, for all of this to work, there needs to be what's considered proofreading. Right? Mistakes do happen even in a complex natural process like this, and so you've got to make sure that that everything you're doing ends up working out the right way. Remember, we don't want mistakes in our genetic code. So DNA has to be faithfully replicated, and so there are about 1 in 10,000 bases the polymerase inserts the wrong nucleotide, which isn't actually that bad. That's a pretty good rate. And so when that happens, they can be removed and then put a new base in. And now other times DNA can be damaged in other ways, like by chemicals or radiation, and there's a mechanism to cut out and replace it with the correct bases. So that mismatching of base pairs can occur at a rate of about 1 in 10,000, and they do try to proofread and repair those. However, there are chances of what's called a mutation, which is about 1 in 100,000, which means that that gene is changed permanently. Okay, and because the human genome is so large, at that rate, the mutations do end up adding up, and basically every one of us, on average, has about 3 to 4 different mutations in our genes. So there's lots of different ways to proofread and repair, and we've got different enzymes and proteins that help fix those sort of things. And so here's some accuracy for you. The chromosome of E. coli contains about 5 million base pairs, and it can copy that DNA in less than an hour. Now there are 46 chromosomes of human cells, and that's 6 billion base pairs. So if you print one letter of that at a time, you could fill up over 900 volumes of Campbell's soup, or you could take a cell a few hours to copy this DNA with amazing accuracy, right? There's maybe one mistake per billion nucleotides. So that's really accurate. So now that this genetic information is encoded, and that's encoded as a triplet, which we talked about last time, and so there's about 64 codons that code for 20 amino acids. Okay, 3 of those codons are stop signals, so things that say, nope, we're done. Okay, and then there's one that's a start. Okay, and this code is universal for all living organisms, so every living thing works on the same code. And here they all are. These are the codon messengers for every single amino acid that our body can make with DNA. So as we can see, here's down here our start code. We've got our 3 stops, and then the rest of these guys are all the different amino acids that make up proteins in our bodies. So cells are going to be governed by this chain of command. DNA creates RNA, which is kind of like an instruction manual for a protein. So in transcription, you synthesize RNA under the direction of DNA, and then translation is where we take that RNA and we turn it into a protein. We're going to stop right here though, and we'll start doing transcription and translation next time. Thanks for watching.